Okay, now we'll focus on proteins, which is one of the biological molecules. So the monomer, or the building block of proteins, is the amino acid, and there are 20 different amino acids. Now, this figure here is an example of a generic amino acid. So this group right here that's shown as R, what this means is everything else about this amino acid would be exactly the same for all 20. This part is what changes for each of the 20 amino acids. So it can be as simple as a hydrogen located here, or it can be a small ring structure, have other elements like sulfur and oxygen. So the side chain, or the R group, is what's going to be different. But when we look at an amino acid, first thing we want to see is it's still carbon-based, right? We still have carbon as part of the amino acid, still have hydrogen present, okay? We still have oxygen, but in addition to that, and all amino acids are going to contain nitrogen. The side chain here can add additional elements. Some of them will have additional elements like sulfur. But all amino acids will have carbon, hydrogen, oxygen, and nitrogen. Now, if we look at the structure, what we see is there are two important functional groups in every amino acid. And the first is this one right here, which you should recognize now as an amino group. So there is a nitrogen with 2 hydrogen atoms. We also have a carboxyl group, which is a carbon double bonded to an oxygen and another OH or hydroxyl group. So that's the carboxyl group. So every amino acid will have these two groups. The way that proteins are formed is through the dehydration synthesis, just like we saw with carbohydrates, where the amino acids are going to be bonded together. When many amino acids are bonded together, it's called a polypeptide. And the reason that it's called a peptide is because the bond in between amino acids is called a peptide bond. We'll look at that a little closer in a second. But what should we know? So polypeptides then, either alone, a single polypeptide can be a protein, or sometimes it takes more than one polypeptide to come together to form a protein. And proteins can serve in a structural way. They can provide structure to cells or tissues. They can also be enzymes. So as, for example, in your digestive system, you have multiple enzymes that are breaking down carbohydrates, proteins, lipids. There are many enzymes in your body that are performing specific functions. Hormones are also proteins that can signal, right, and serve as communication molecules that travel through your bloodstream. Now let's look a little bit closer at, I mentioned to you that the bond between amino acids is called a peptide bond. This is a covalent bond, is a strong bond. And this right here is an example of a peptide bond. So we have, starting out, amino acid 1, right, and amino acid 2. Now this right here, I'm actually going to write an R instead of a hydrogen. So in other words, this could be any generic amino acid. That R group can be different depending on the amino acid. So just like with the rest of the biological molecules, when the monomers are put together, there's a molecule of water that is lost through this reaction, and a new bond is going to be formed there between those two amino acids. And this bond is called a peptide bond. Notice that it is between the carboxyl group of one amino acid and the amino group of the neighboring amino acid. So that's a very specific type of bond, and we only find that between amino acids. If this were a polypeptide, right, we would have continued, there would be another amino acid here, right, and then it would continue on with a chain of amino acids. Now, to the right, I have given you 4 examples. Remember, there are a total of 20 different types of amino acids. Alanine is one of those. So here's the amino group. Here's the carboxyl group. This is what the R group looks like for alanine. So for valine, we have the amino group, the carboxyl group, and this is what the R group looks like. So this is just, right, this R group is just carbon and hydrogen, or hydrocarbon. This R group is only carbon and hydrogen, or a hydrocarbon. However, we see with lysine in the R group that there is another nitrogen, and we see with aspartic acid that there is another carboxyl group that's added. So the R groups can differ between amino acids, and that plays a role in how the protein will ultimately, the structure of it will be, or how it will be folded. So proteins have what we call 4 levels of structure. And the 3-dimensional shape, or the way the protein is folded, is very important to its function. So the very first level of structure, or the primary protein structure, is just the sequence or the order of the amino acid. So in other words, which amino acid comes first? Is it alanine, and then valine, and then glycine? What order are they actually bonded together? And what determines this order is actually found in the DNA, or in your genes. In other words, the gene is the instructions that say, this is the order of the amino acids. And later in the semester, we will find out exactly how this process is carried out. But as you see here, these look like beads on a string, right? Each of these beads would be a particular amino acid. And the order of those is determined by the gene, or the DNA. Now, the secondary structure of a protein is what we'll call local folding. In other words, based on what amino acids are present in what order, this will cause, just based on the kind of chemistry that's present there. In other words, are some of those side chains, or those R groups, hydrophobic, in other words, water-fearing? Are some of them hydrophilic, water-loving? Are they repelling one another? Are they attracted to one another? All of that plays a role in how the local folding will happen. Some examples of local folding are, it may form this spiral structure, right, an alpha helix, or it may form something called a pleated or beta sheet. And usually this is sort of a repeating pattern for a particular area, right? And then you may have an area with a sheet and an area with a helix. Now, the next level of structure or folding is the tertiary structure. Basically, this is looking at the entire polypeptide, the whole 3-dimensional structure. So if we look at this here, what's pointed out is maybe this area here is a pleated sheet, right? Or this area here is a pleated sheet, but this may be an alpha helix. But the whole entire polypeptide, how it's folded together, the whole 3-dimensional shape, is the tertiary structure. Now, if the protein actually has more than one polypeptide chain that comes together to form the functional protein, then it will have what's called quaternary structure. And basically what that means is, how are those different polypeptide chains, once they have a 3-dimensional tertiary structure, how are they associating together to form this protein? Now, as I mentioned, the 3-dimensional shape of a protein is very important in its function. The way that it recognizes the molecules that it binds to is based specifically on the way that the structure is folded. But the folding of the protein can be, it can become misshapen because it becomes denatured. So proteins, remember, are smaller than cells. So the cell is the smallest unit of what we consider living. So proteins are not living, right? They're biological molecules. But they can become denatured, or what that means is they don't have their functionality anymore. And some of the common things that will cause them to be denatured are a change in pH. So the pH of a solution affects the way that the amino acids interact with each other, so it affects the protein's folding. Temperature can also affect the folding of a protein, as well as salt concentrations. So these are some examples of what causes a protein to become denatured and no longer have its functionality. Now, another thing I want to point out to you is that we can have mutations in genes. So as I mentioned, a gene, or the DNA, is what is instructing exactly the order of the amino acids to be added in the formation of the protein. So hemoglobin is an example of a protein. This protein is found in your red blood cells, and its job is to carry oxygen throughout your bloodstream to all your cells and tissues. It actually has 4 separate polypeptide chains, or what we call polypeptide subunits. 2 of them are called the alpha version, and 2 are called the beta version. Now, in people who have sickle cell anemia, what happens is there is a mutation in the gene for hemoglobin for one of the for the beta chain, and this mutation affects only one amino acid out of the entire polypeptide. And this on the left side would be, this is just sort of a small, we're looking at a few amino acids out of the entire subunit, but what I want you to notice is glutamine would be the normal amino acid that should be coded for here, that should be added. But instead, in folks who have sickle cell anemia, the gene for that has a mutation, and it codes for the amino acid valine, which causes this polypeptide to be folded differently, and it causes an error in the hemoglobin molecule so that it doesn't carry the oxygen throughout the bloodstream like it should, and it actually causes the red blood cells to be misshapen. So that one change in amino acid can have a very large effect on the folding of a protein.